It may be different at like school. I really have to do
It's only one. We didn't watch.
Okay, so we're going to begin today with a short activity to remind ourselves about where we left off with homeostasis. So what I'm going to ask you to do is really simply just draw this process of negative feedback. So giving you an example. So heat exhaustion happens when the body is really trying hard to return body temperature to its set point, working too hard for that. So we want to draw the normal negative feedback control for temperature. So I want you to think about when we're controlling temperature, what our regulated variable. We want to think about what the set point would be. You're going to draw that on your little graph you're going to make. Then you're going to mark down what the error signal is. Just to make sure that we understand these three terms that we went over on Friday. I'll give you a second to do that, and then we'll drop it down. Okay, so beginning with this graph, wants to let me know what the regulated variable is here in this problem. Exactly, our regulated variable here is just the temperature. And what axis am I going to put the temperature on? 50-50 that. Yeah, exactly. Our regulated variable will be on the y-axis. It's temperature, not a trick question. Great. Okay. What about time then? Where's time going to go? X-axis, perfect. So when we're thinking about maintaining homeostasis and negative feedback loops, we're looking at a variable over time. All right. And where am I going to put our set point, X or Y? Yeah, I heard someone say it. We're going to put that on the y-axis. So our set point will be here. Okay. So as we are measuring temperature over time, we'll see some fluctuations. So we might see the temperature go up Now, I don't know how to ask this as a question. So where am I going to draw a dotted line for our error signal? I'm going to draw it at the bottom, or am I going to draw it at the top of this? Yeah, exactly. We're just marking our error signal as the difference between our set point and our actual measurement. Then we have our negative feedback is just pulling us back down to that set point. Or we could have the opposite. We were thinking about heat exhaustion, so we are thinking about temperature going up. But if we were thinking about hypothermia, or maybe when you sweat and get too cold, we would still have an error signal downward. This would still be negative feedback because we're just pulling in the opposite direction versus our change. So the negative here is not about plus or minus, it's just about opposite. That is what we talked about on Friday. So a couple more reminders. So when we think about these negative feedback loops, we can draw our actual regulated variable and what's happening to it. But as we're thinking about physiology, we also want to think about why that's happening, what the processes are here, what organ systems might be involved. So we also want to think about pathways. So when we think about pathways here, we're kind of thinking about like nervous pathways, like the nervous system. So whenever we have a negative feedback loop, we're talking about regulating that variable. We're going to note that part of our pathway will be first sensing a change. So we'll have sensors in the body detecting the change. So these will usually be some sort of receptor on some sort of cell, most likely. Then we'll go to the integrating sensor. So we will have some signal traveling up a nerve, likely to your brain. So to our integrating center, which is going to assess that change and figure out what response we need in order to correct it. So then we have a signal going out from that integrating center. So again, think something traveling along a nerve to an effector organ. So different types of muscles, different types of glands, which are then going to allow us to adjust the error signal, adjust what has happened to that deviation from the set point. So that's all a negative feedback loop is. We have an error signal telling us there's been a change. We sense it with a sensor, send that signal to an integrating center, and then we have an effect on that variable through our effector organ. So that's what we mean when we say pathway. So negative feedback loop is always just in the opposite direction than our change. We're decreasing the error signal. So what this means by making the change from the set point smaller, all that really means we're bringing ourselves back towards the set point. So that dotted line that I drew would get smaller and smaller and smaller as we get closer back to the set point. So this is continuous. So it's not a huge problem if you have a little bit of an error signal from the set point. Your body is designed to be able to make these adjustments. Changes happen to things like blood pressure all the time. We went through an example as you stand up, right? So your body is supposed to be making these fine adjustments to stay in homeostasis all the time so that your different cells, your different organ systems have a predictable environment to function in. Because as we will see, a lot of our concentrations of things outside cells, different pressures, are going to make a difference to how cells and our organs function. So we need to have a predictable environment. So here's the exact example for body temperature. So we have our typical body temperature, our normal body temperature of 37 degrees Celsius. As we have deviations here, so the brain can tell based on sensors that either we're getting too hot or too cold. We get too hot, we're going to send out a signal to effector organs, in this case, our sweat glands, telling us to cool down the body. So those are our gland, our effector organ here for adjusting temperature. And then once we cool down, those sensors are still sensing what's the temperature of our body. So they're going to send that signal continuously to the integrating center. And eventually our integrating center is going to know, oh, okay, we're back at our set point, and those sweat glands will get turned off. That's basically what inhibited means, right? Like if you have a lot of inhibitions, you don't do things. Same thing when we're talking about physiology, when we're talking about these loops. When we say something is inhibited, we mean we're like basically turning it off so it doesn't do whatever it was doing. So this is the whole process of regulation. When we talk about antagonistic effectors, what we mean is that sometimes we have something that's going to cool down the body, and sometimes we have something that's going to heat it up, right? We might want to move this needle in opposite directions. So that's what we mean by antagonistic effectors. They have the opposite action on our regulated variable on our set point. So for cooling, we have our sweat glands, right? So that's a gland, that's an effector organ that cools us down, brings us back down to the set point. But if we get too cold, that response of shivering or even your hair sticking up, that's also an effector organ, in that case, a muscle, right? Those muscles are actually generating heat as you shiver. So that brings us back up towards the set point. So we maintain homeostasis in both directions. Both directions count as negative feedback. And I keep repeating that because sometimes people just get confused about that word negative in there. So that's why I'm really hammering you over the head with that. So we'll see similar things happening with blood glucose levels, blood pH as we move through this course. When you see diagrams about negative feedback in your book, they may look something like this, and this is an example that we will run through later on. So what we are seeing here, right, is we're going to see an action likely up at the top, depending on exactly where they start the diagram. So here we see that when we eat food, this raises blood glucose. So this raises the sugar in your blood as you ingest food, pull it across your intestine walls and into the bloodstream, so into that plasma, into that specific type of extracellular fluid. Then we have a reaction by a specific gland. So in the pancreas, as we have a receptor or a sensor that notes that raise in blood glucose, we have that process by an integrating sensor, and then we have the effect of an effector organ, in this case, those pancreatic glands, and they produce something called insulin that you have probably heard of, right? So super important in diabetes management. So as we have a rise in blood glucose, we have a gland that produces insulin. And what insulin does is it increases cells uptaking glucose. So when there's insulin present, cells pull glucose into them. Because plasma is extracellular fluid, once that glucose is in a cell, no longer in the extracellular fluid, so it's no longer in the plasma. So since it's not there anymore, the blood glucose has gone down, and that's how we regulate the glucose level of our blood. Now we also see here on the left-hand side that this is a negative feedback loop. That's what this minus sign means. Okay, so this means that now we could pull this all the way back to the beginning, right? So we see that we had an opposite action. Raise in blood glucose ultimately causes this decrease in blood glucose. So if we then wanted to see what happens with the decrease in blood glucose, we'd reverse the actions of all those arrows, basically. So decrease in blood glucose would mean that our insulin levels are lower and we'd have less cellular uptake of glucose. We have a corresponding process, an antagonistic effector called the glucagon on the right here. So we don't have to run through that right now. We'll have a whole unit where we focus in on insulin and glucagon and blood glucose. But that's how you're going to read these diagrams. So pay careful attention to the arrows. And when we see these negative or plus sort of dotted lines on the side, that's telling you how do we turn off this loop, or very occasionally, how do we speed this up so this happens more and more. Okay. So positive feedback loops are much more rare. So when we think about a positive feedback loop, you want to think about making a snowman rolling a snowball bigger and bigger and bigger, or a snowball going down a hill, right? If we were to graph a positive feedback loop, it's just going to go up and up and up, right? Positive feedback. Someone says to you, yes, do more of that. So you do more of that. Goes up and up and up. And that's fundamentally really all that positive feedback means. That when we see a change, so say we were to see what we might have thought of as an error signal in a negative feedback loop, instead of pulling ourselves back down to the original point, we increase that change. We make it bigger and bigger and bigger. So that's why it's a positive feedback loop. Okay. At the end of a positive feedback loop, we will eventually turn it off with a negative feedback loop. So we'll see positive and negative feedback loops working in sort of concert when we look at specific processes in the body. So if we're thinking about a positive feedback loop, we might say that the end product of the process, so whatever we're making at the end, whatever hormone, for example, we're making, it also starts the process. So the presence of that hormone causes more production of that same hormone. So we're amplifying an effect with positive feedback. So this doesn't work alone because you don't want your whole body to be made of that one hormone, right? That wouldn't work. So eventually there will be negative feedback loops involved here as well. But when we're looking at various physiological processes, we may see either of these types of feedback loops. We'll focus on these positive feedback loops really mostly when we talk about blood and when we talk about the reproductive system. So the most common examples of positive feedback loops are when we form blood clots. So you don't want just two little platelets to stick together. You want to actually seal over a cut. So that means that in your body, when you start to form a clot, that's actually a signal to make that clot bigger and bigger and bigger until you have actually healed your wound. So that is our positive feedback loop there with clotting. Another example is in uterine contractions during childbirth. They get stronger and stronger and stronger until birth occurs. So here's an example of what that might look like in your book. So the pituitary gland, the gland sort of hanging on your brain that causes the release of this hormone LH. So LH stimulates the ovaries to produce estrogen, and then the presence of that estrogen in certain conditions produces more and more and more LH secretion. And this is actually how ovulation works. Positive feedback loop will appear multiple times when we get to reproductive, which will be at the end of A and B2. So most of the things we're going to talk about for the first part of the course are going to be negative feedback loops, but don't forget that positive feedback loops are possible, do exist, and will be important for certain processes. Now, one side note, this pathway that we've been talking about has really been following the extrinsic pathway of regulating a specific regulated variable. So I keep talking about the integrating center as we can think about the brain as being an important integrating center. So that has been part of our pathway. But we will also see that some things are regulated intrinsically. So right there on location where the cell is. Sometimes that response is going to be really, really local, and we're not going to have to go out to the brain to create a response. We might have that cell or the wall of that blood vessel respond directly without sending some complicated signal to some other organ system. As we go through our other units, we will see these words again, intrinsically versus extrinsically. When we talk about regulation, we are often going to be teasing apart, okay, what happens at the local level, what are immediate sort of responses that don't require nervous system. They're often going to be faster when we're regulating things intrinsically because we're right there. And they're only going to affect that local area. Whereas when we regulate things extrinsically, that's always going to be something coming from outside. So when we see this word extrinsic regulation, which we'll see regularly, we're often going to be thinking about the involvement of the nervous system, sort of longer, more global processes that may affect multiple parts of the body at once. Questions at this point? This is basically our full preview of homeostasis and introducing some of these concepts that we'll mention as we move through very short. This is kind of like a preview, getting these terms down before we dive into very specific examples. So the next thing we're going to move on to is biomolecules and enzymes. So hopefully you all spent a little while looking over biomolecules over the weekend. That was part of our mastering A&P assignment for today. And hopefully you have all seen biomolecules before in your previous courses. Now, the reason we're talking about them now, we all want to get on the same page about these biomolecules and enzymes. What we're going to be trying to focus on is the properties of specific biomolecules, so how they interact with each other, and especially we want to keep our mind on how they're going to interact with the environment. So we want to think about how would they interact with a cell membrane, how would they interact with extracellular fluids, like fluid outside the cell, how would they interact with regards to the cytoplasm, to that intracellular fluid. So those are the key components we want to keep in mind as we think about these molecules for the purposes of physiology. We want to be thinking about how they're going to function in the body. So when we say biomolecules, or we pretty interchangeably use the term macromolecules for the purposes of this course, we're talking about molecules that are synthesized, well, made by living things. They're primarily going to contain carbon. They're going to be carbon-based molecules. They're going to be relatively large. We'll see other atoms involved in our biomolecules, frequently oxygen, hydrogen, and nitrogen. And when we're talking about our phospholipids, phosphorus will also be important. So what we want to remember about carbon, a little sort of cheat sheet, carbon forms 4 bonds. So we're going to see things centered on carbon molecules. Okay. And we're going to break down our biomolecules into 4 types. So we're going to talk about our carbohydrates, so things like sugars, starches. We're going to think about lipids, basically fats, oils. Some of our hormones will be lipids, and critically, our phospholipids that are the building block of our cell membrane. Think about proteins, which are going to do most of the actions in our cells. When we think about proteins, we want to be thinking about receptors embedded into membranes. Some of our other hormones will be protein-based hormones. Our proteins are going to have a lot of effects on these different systems in the body. And finally, we're going to remember that nucleic acids, DNA and RNA, are their own type of biomolecule. So if we look at a carbohydrate tells us what it is in the name, right? We're going to have carbon, oxygen, and hydrogen. So we're going to see carbon and a bunch of OH groups, right? These OH groups are in the name. We're going to see them in ring form in the body. So we can see a carbon ring with a bunch of these OH hydroxyl groups on the side. We can name them in order of how large they are. So a monosaccharide is just one of these rings. So our monosaccharides here are glucose. This is our most important. We'll talk about glucose a lot. These others we won't talk about all that much. But glucose is a monosaccharide, so just one unit, one monomer of a carbohydrate. We have disaccharides. So di meaning 2. The saccharide bit, like saccharin means sweet, means sugar. So that would just be 2 of these monosaccharides linked together. They don't have to be the same kind. So we might have glucose, our super important monosaccharide, linked up to a fructose. which is like fruit sugar, to form sucrose, which is table sugar. Okay. And then we'll have polysaccharides. We're not counting exactly anymore. Poly meaning many, saccharides still meaning sugar. Okay. So here we have an example that we'll talk a lot about, glycogen. So glycogen is a way that your body stores glucose in chains, right? So each of these little circles is a glucose unit here. So you can see we've linked them all together. So we're going to see glycogen storing glucose in places like the liver and some skeletal muscle, so that we can then release these glucose units later. The next topic we're going to talk about, our next PowerPoint, is metabolism. So we're going to be thinking about cellular respiration. So we're going to be thinking about breaking down glucose. It's going to be a critical topic for understanding how the body makes energy. So this is a tidbit for the future. So we have these glucose molecules stored in glycogen in animal cells. So when we're thinking about carbohydrates, we might want to think about how we build up these longer chains. So like, how would we store glucose in this bigger glycogen molecule? Or we might think about how do we break up carbohydrates, and we'll think about that, especially when we think about the GI system, when we think about digestion. So we are linking together our carbohydrates with covalent bonds. So they're stuck together pretty permanently. So when we link them together, we do this as part of a condensation reaction. So what that means is when we take two monosaccharides, so two of these individual monomers, these single units, when we link them together, we're going to get water as one of our products. So we take these two groups, we're going to make a covalent bond with oxygen in the middle. So we've peeled off one of our hydrogens and a whole hydroxyl group here, just leaving the oxygen in the middle. So this is pulled away as a water molecule. So condensation, like condensation on the outside of your water bottle, we've formed these little water droplets. So that's how we link them together. This means that we can break them apart in the opposite way. So if we add water, we can have hydrolysis reactions. You'll see that in the body, we speed that up with various enzymes. But it is true that if you add water to these sugars, we can break them back down, adding that water back in, and get our original monomers. So hydro, meaning water, lysis, meaning breaking things down. So to lyse something is to break it apart. One last thing to note about the carbohydrates, which isn't on this slide, but I want to provide this as a preview to you before we move forward ultimately into the cells. So the presence of these OH groups, these hydroxyl groups, this is going to make carbohydrates polar molecules. So our polar molecules are going to be attracted to water, basically. And they're going to be repelled by our cell membranes when we ultimately get them. So that's one of the things that we want to pay attention to here with our carbohydrates. I'm saying that now because that separates them from lipids. Lipids are the opposite. So a lipid is going to look really different depending on what lipid we're looking at. So we're not going to always see that very same typical ring structure, which is a sort of diagnostic characteristic, something very familiar about carbohydrates. A lipid is really defined as something that's just mostly made of hydrogen and carbon. Not a lot of oxygen, and we'll see a little bit. This presence of tons and tons of hydrogen and carbon alone makes our lipids non-polar, meaning that they are also going to be hydrophobic, so they're going to repel water. We pay attention to these relationships to water because as we were breaking down our total body water, our body fluids last week, right? The environment that our cells are in the body is primarily water. So how these molecules interact with that water is really critical to how they function physiologically. So our lipids recognize them because we just see tons of hydrogen, tons of carbon. They repel water, they're nonpolar. So there are 5 classes of them that we're going to see in the body. So we have triglycerides, which is our most familiar example. So one of the key ones to be able to sort of visually recognize, and it's going to hammer home this hydrocarbon concept pretty well. So those are fats and oils. We also see ketones, phospholipids, which are going to be the building blocks of our cell membrane. So we'll spend a fair bit of time on those as well. Eicosanoids and steroids. So steroids are going to be part of one of our classes of hormones. We have steroid hormones. So we'll spend a little time looking at that structure as well. So hopefully you have seen triglycerides before. So triglyceride, again, tells you what it is. So tri is telling us that we have 3 of these fatty acid chains. And then glyceride is telling us that we have this glycerol backbone. So we have this 3-carbon molecule. I always think this kind of looks like a flag, right? So we have this 3-carbon molecule as our backbone. And then these fatty acids are really what makes this a lipid. As you'll notice, we have some oxygen here in that backbone, but then we just have carbon and hydrogen all the way down the line. And that's basically it here. So these in yellow, these 3 fatty acids are really what's making this molecule a lipid. Our 3 fatty acid chains. Those long carbon acid chains can either come in saturated or unsaturated. And what that's telling us is basically how much hydrogen they have bound in. So each of these carbons, right, every carbon takes 4 bonds. So we know since it's a chain, we have a carbon on either side, right? But we have 2 more bonds that carbon in the center is going to need to make. So we can just have a hydrogen on either end. That's super simple. Those are going to form our saturated fatty acids. They're saturated with hydrogen, so as much hydrogen as they can hold. But we don't have to fill out those 4 bonds that way. We can also form a double bond with one of the neighboring carbons. So that would be bond 3. And then we still have a hydrogen on one side. So this is unsaturated. This is what an unsaturated fatty acid is. The reason it's unsaturated is that we could fit another hydrogen in there if we hadn't formed that double bond. So that's all that means. And when we're thinking about unsaturated fatty acids, sometimes when you're reading about what's healthy, unhealthy, you'll be looking at those unsaturated fatty acids and they'll specify whether they're trans fatty acids or cis fatty acids. And what that's telling you is about the position of the hydrogens on either side of that double bond. Okay. So here, if on either side of that double bond, those hydrogens on those 2 carbons are on the same side, we have our cis fatty acids, so cis same. But if they pop off on either side, so on opposite sides of that double bond, that's a trans fatty acid. So our trans fatty acids, because of that shape, may behave a little bit more like saturated fatty acids because they don't form the same kind of bend in the chain. So that's why that's sometimes relevant from a health perspective. Moving on to our ketones. We'll think less about these, but we will think a little bit about them when we think about fat metabolism. And if you think about diabetes as a disease, so in diabetes or in other low-carb diets, like the keto diet, right? What happens is that we're breaking down triglycerides, and we're breaking down those molecules we just looked at in adipose tissue, so in the fat. So we're breaking down triglycerides, and this releases ketones. So the sort of byproduct of the process of breaking down the triglycerides gives us ketones that go into the blood. And they're basically processed by the liver here. Those fatty acids that we're breaking down off of the triglycerides are getting converted into the ketones in the liver specifically. If we have a buildup of these ketones, you can actually smell it on both breath. So we call ketosis or ketoacidosis as the presence of high numbers of these ketones in the blood. And it has a sort of, I call it sort of sweet, right, acetone breath. So this is a sign of diabetes or a possible sign of diabetes, or just a sign that someone is in ketoacidosis, that you can actually smell some of these molecules. So our next lipid, remember all these lipids, they look different, but what we're focusing in on is that they have a lot of hydrocarbons, so lots of hydrogen, lots of carbon. They're going to have at least one part that is nonpolar, hydrophobic, repels water. Our phospholipids are sort of special because they have two parts. They're going to have a part that's nonpolar, but they have an extra phosphate group tacked on, which creates something called a polar head that is going to be hydrophilic. So for this reason, we think of our phospholipids as being amphipathic. So they're both attracted to water and repelled by water, depending on which end of the molecule we're paying attention to. So if we draw it out in sort of like biology terms, we don't write in any of the atoms. We just kind of focus on the shape. So our polar hydrophilic head that's attracted to water is here. So at one end, we have this hydrophilic head. And then the nonpolar end, link that up. So this is our polar head. When something is polar, it's hydrophilic. So attracted to is that philic part. Water is the hydro part. So that's on one end. And on the other end, you can see how this kind of looks like the fatty acids on the triglycerides. That's because that is actually what's in the squiggle. So this is just carbon and hydrogen in these fatty acid tails. So they are nonpolar. They're nonpolar, and this makes them hydrophobic. So basically they repel water. So hydro, water, phobic, fear. So water fearing. So this gives phospholipids a really sort of special property. They're lipids because of these nonpolar tails, because they're mostly made of hydrogen and carbon, but they also have one end that's attracted to water, which means that they're going to form really specific orientations and really specific sort of structures when they group together in a watery environment. And that's what allows them to form the basis of our cell membrane. So if you were to have just sort of a handful of phospholipids and you drop them in water, there are some different structures that they could make. So one we won't talk about much, we'll see this come up once when we talk about digestion, but you might have seen it in sort of face cleansing products. It can form something called micelles, which is just all the fatty acids towards the center. This isn't that helpful for us when we're thinking about cells because there's no room in the middle, right? All those fatty acid tails, all those hydrophobic, non-polar parts of the molecule are just touching each other in the center. So the type of structure we really care about when we think about phospholipids are our phospholipid bilayers here in figure A. Surfactant is another type of phospholipid that we'll see when we get to lungs, but for right now, we really just want to focus on these phospholipids that are forming the bilayer because we're going to be thinking about the creation of our cells. So it's happening here, right? Inside the cell, we know we have this watery substance called our intracellular fluid, which is mainly going to be composed of our cytoplasm. So this would be inside the cell. On the other side, extracellular fluid. So probably interstitial fluid if we're thinking about just some random cell, but might be plasma if we're thinking specifically about a red blood cell. Okay. But both the ECF and the ICF contain lots and lots of water. That's what they're primarily made of. So when we're thinking about phospholipids, they're going to arrange themselves so that the part of them that likes water, that hydrophilic part, is touching water, right? So that's attracted to the water in the ECF, in that extracellular fluid. And it's also attracted to the ICF, that water in the cytoplasm, essentially. Now, it's important to our phospholipids as well that these nonpolar tails, these fatty acids, this hydrophobic part of the molecule, it absolutely does not want to touch water. So they line up end to end with those fatty acid tails touching each other towards the center. So a bilayer, right? Bilayer, bi means two, layer is literally just a layer, right? We have a layer of two of these phospholipids, but they're arranged so that they're in the opposite sort of configuration, right? So end to end, we have polar hydrophilic heads on the outside, and on the very, very inside of the bilayer. But in the center of the bilayer, we have two fatty acid chains here pointing towards the center, and then these two fatty acid chains pointing to the center from this second phospholipid. Questions about this arrangement, because we eventually move into cells. It's going to be different.
How are the phospholipids put together? Like the bilayer and then the.
So how are they pulled together? Is that what you said? Yeah, so it's actually just a reaction of how these molecules are responding to the presence of water. So it's really just that they're like running away from the water in the environment. So they'd be moving around until they reach this configuration, at which point they're stable and no longer trying to sort of spatially escape each other. They're not like linked together by a specific force. They're not covalently bonded to each other or anything. It's just this is the arrangement that has them, you know, otherwise they're kind of like repelled like magnets from the water, essentially. That's a great question. Okay. So this arrangement is why when we look at our biomolecules, one of the things we really want to focus on are those polar versus nonpolar, hydrophobic versus hydrophilic properties. Because aside from the fact that we want to understand how this membrane is forming, we're going to be focusing a lot on, okay, what can go through that membrane? What can go through, what can't go through, and why? So when we think about that, we're going to be focusing on particularly what can mix in with these hydrophobic nonpolar tails. Because as you can see, that forms like such a thick layer, right? Like spatially, that's the bulk of what's in this membrane. Okay, so we do have a little bit of polarity at either end arranged near the water. But if you were trying to drive through a membrane, you'd have to cross through a lot of nonpolar region. Oh, that's our previous thing. So our next category of lipids are our eicosanoids. So these are really just modified fatty acids. So they're kind of bent in half. We won't see them much in this class, but they are important in the future in sort of intracellular communication. So as cells are sort of signaling to each other, one of the ways they can do it is through these molecules. So examples that we'll maybe see are prostaglandins and thromboxanes. I wouldn't focus too much on these or their structure other than that they exist and are an example of a lipid. So the way we could recognize that, right, is just that they're mostly made up of carbon and hydrogen. We see carbon and hydrogen all over. We see a little bit of oxygen, right? But we don't see very many of these hydroxyl groups. They're not in this typical ring structure that we expect to see things like glucose. So they're mostly going to have these sort of non-polar traits, except at certain locations. Steroids we care a little bit more about. All steroids are derived from cholesterol, so they're all going to have this same sort of 4-ring structure in the center of them, but they may have other pieces kind of hanging off. So steroids are going to be a class of hormones, which is why we care about them mainly. But cholesterol itself is going to be an important part of cell membrane. So examples of steroids that we'll see in the future are things like estrogen, testosterone, and aldosterone. So those are some of our hormones. So some sex hormones, and then one that we'll see more involved sort of with the renal system. So the way we recognize them is that we have these 4 rings pressed together, all made up of carbon and hydrogen. So if you don't look carefully, they might look a little bit like a carbohydrate, but you'll notice we're missing all that oxygen, missing the hydroxyl groups, and our carbohydrate rings are separated out with an oxygen in the middle, whereas these rings have 2 joint points right here, right? The ring itself is stuck onto another ring. Final topic is proteins. Okay. So our proteins are going to be big, long chains. When we say protein, we're really thinking about a polymer. So a big set of amino acids covalently bonded together. An amino acid, we have 20 different kinds, are going to be our monomers, our building blocks of these proteins. I'll spend a lot of time thinking about different types of proteins in this class and their shapes. So we definitely won't be getting to enzymes until Wednesday, but enzymes are going to be one of our types of proteins, right? Enzymes are made of proteins. The basic structure, and we will pick up here later, but I want to just point out the first little bit. Our proteins are going to be built of these amino groups, or of these amino acids chained together with peptide bonds. So an amino acid has a central carbon, which we know needs 4 bonds. I'll pick a different color to highlight that. So this carbon needs 4 bonds. 1 bond is just to a hydrogen on the bottom. On one side, we have our carboxyl group. So our carboxyl group is a carbon double bonded to an oxygen, and then this hydroxyl alcohol OH group off to one side. And then on the other side, we have our amine group, which is a nitrogen bonded to some hydrogens. And the part that makes our 20 amino acids different from each other is this R group. So the R group is going to vary between our 20 amino acids, and that's ultimately what's going to help give our different proteins different functions. So we'll pick up with thinking more about proteins on Wednesday.